welcome everybody to the May edition of the IIDR Rounds. I'm Laurie Burrows. I'm the interim director of the IIDR, and it's my pleasure today to introduce our two speakers. The first speaker is Dr. Kevin Woodward. He is an associate professor of medicine in the Division of Infectious Diseases at McMaster. Over his career, he's worked in inpatient infectious diseases, conducted clinical trial work in the areas of HIV and sexual health, and has been involved in provincial and national projects for HIV prevention. And today he'll be discussing gonorrhea treatment, guidelines, and resistance data. Our second speaker is Gerry Wright, who needs no introduction to you. He is a professor of biochemistry and biomedical sciences at McMaster and a leader in the fight against antimicrobial resistance. He is known for his role as the scientific director of the Michael G. DeGroote Institute for Infectious Disease Research, but recently has pivoted to launching Canada's global nexus for pandemics and biological threats, of which he is the inaugural lead. And today he'll be discussing new gonorrhea drugs in the pipeline, as well as the discovery of others. So thank you both for talking to us today. We're going to be hearing about the rise of a new superbug.
Thanks, Laurie. So this slide is because yesterday was May the 4th, but also because Dr. Kornica requested that after he saw the title that I do the rise of super gonorrhea. So I'm going to talk just really briefly on the historical perspective of gonorrhea resistance, and then review some epidemiology and talk about gonorrhea resistance in Canada, and then talk about the new CDC guidelines. And really this came out of in December of 2020, the CDC released new guidelines where they changed the recommendation for gonorrhea treatment. Traditionally, it was a dose of ceftriaxone at 250 milligrams plus a gram of azithromycin. And they've updated those guidelines in the States to be a single dose of 500 milligrams of ceftriaxone. Canada hasn't changed any of their guidelines yet, but I've been getting a lot of questions from people about what should we really be doing for gonorrhea? Do we need to go to 500? And so I thought this was an appropriate topic to review. I think the other thing that is apropos of the time is that even with COVID, we haven't really seen that much of a change in STI rates. And in fact, some of the STI rates have remained the same or going up. So despite there being a lot less testing that's actually happening. So these epidemics have not gone away despite our current pandemic. So this is just a picture of a mailbox from the 40s, and this is sadly no longer true. But it used to be penicillin cures gonorrhea in 4 hours. See your doctor today. We can give you a shot and get rid of it. And sadly, penicillin has gone away for the treatment of gonorrhea now. Gerry has a much better picture than this. I took this from a 2011 article. But basically it goes over the timeline of antibiotics that we've used for treating gonorrhea. So you can see back in 1897, they actually used a silver compound. So, I think that probably didn't have a lot of efficacy. And really, once they got sulfonamides in 1936, the use of the silver compound for gonorrhea went away. But what you can see here is that very quickly, there was widespread resistance to the sulfonamides. So even though that was introduced in 1936, by 1944 to 45, there was widespread resistance. Luckily in 43, they had discovered penicillin, and that became the mainstay of treatment. But if we fast forward to the late 50s, we were starting to see penicillin and streptomycin clinical resistance. In the 70s, they reported beta-lactamase plasmids and high-level penicillin resistance. And so penicillin became much less useful for the treatment of gonorrhea. And then in the 80s and to 83, we had ceftriaxone, cefixime, and azithromycin. However, we began to see resistance to those antibiotics quite quickly as well. So in 85, there was tetracycline resistance that was reported. And then in 1989 was when penicillin completely disappeared off the US guidelines and ceftriaxone was now recommended. By the early 90s, there was widespread Quinolone resistance. By 99, azithromycin resistance. And in 2007, fluoroquinolones were no longer recommended as treatment for gonorrhea. And we'll see this play out in some future slides of what happened in Canada. And then finally in 2011, this was where really people started talking about Superbug with a lot of infographics, you know, of the bug with capes. There is a high-level ceftriaxone resistant strain that was found in Japan, and that was fully characterized. And then the real question is, when are we going to get to the point of untreatable gonorrhea? So a little bit about epidemiology, and this is going to be specific to Canadian epidemiology. So first of all, just looking at STI rates in Canada. So if we compare between 2008 and 2017, what you can see here is that all STIs are rising. And when we had looked at this data previously, there was a question of, well, how much is because we're using gnats instead of cultures, and perhaps they're more able to pick up organism. But really in this time frame, our testing has not changed significantly, but we're seeing a significant increase in these infections. So chlamydia, and specifically the blue line, if you look at gonorrhea, we've had quite a large rise, with gonorrhea rates increasing by over 100% between 2008 and 2017. Just looking at Canada as a whole, you can see here the rates of gonorrhea cases by province and territory, and this is per 100,000. So in Ontario, we're sitting at about 55 cases per 100,000. In 2017, you can see that there's places in Canada where there's much more of an epidemic of gonorrhea. So in the Northwest Territories, Yukon, their rates are incredibly high, and probably some of that has to do with lack of access to healthcare and lack of access to timely treatment. So there's a lot more spread of gonorrhea happening from person to person in those areas. So overall, gonorrhea rates have really almost doubled in the past 5 years. Some trends that we see in the epidemiology, so rates are much higher amongst males and also are increasing faster amongst males than they are amongst females. And in 2017, the rate of gonorrhea amongst males was nearly twice as high as those in females. And most cases are reported between those who are 20 to 29 years of age. And the rates are increasing the fastest amongst those who are 30 years and older. So again, just a graphic representation of what we see. So this is the total number and sex-specific rates of reported gonorrhea cases in Canada. And this goes between, again, 2008 and 2017, which is the latest published data. And what you can see here is that the total rate in the green is going up, but in the blue, the male rate is increasing at a faster rate than the female rate, with our highest numbers being in 2017. So I wanted to move on and talk a little bit about gonorrhea resistance and back to some of the history. So this is antimicrobial data from the 90s. And they looked at antimicrobial susceptibility of gonorrhea strains tested in Canada in 1998. And this picture is looking quite good. You know, our penicillin resistance rate is around 11%. Tetracycline's still high, but things like Cipro and Azithromycin seem like very good treatment options given that low resistance rate for gonorrhea. However, when you zoom ahead just a few years, so this is an article that was published in 2009, and looked at the Quinolone resistance rate in gonorrhea in the previous few years. And what you can see is that from 2002 to 2006, that rate went from about 4% up to almost 30%. And this is really the data that spurned the idea of getting quinolones off of the treatment guidelines. So if a third of the patients you're going to treat with a quinolone will fail because of resistance, then really it should no longer be in those guidelines. And it was removed, I think, in 2007 from Canadian guidelines. So quinolones are no longer on the guidelines now, and really we're looking at back to beta-lactam antibiotics in order to treat gonorrhea. So if we look at all antibiotics within Canada and everything we use to treat gonorrhea, what you can see is that between 2000 and 2008, all of those antibiotics increased the rates of resistance. So whether it's penicillin, erythromycin, the Cipro data we sort of presented before, and Azithro, they're all increasing in over time. However, between 2014 and 2018, we saw a significant increase in specifically azithromycin resistance. So that rate, which was back in 2008, 2009, around 1 to 2%, by 2014 was about 3%. 2017 was a very bad year. It looks like there was a lot of resistance that was reported in that year, but we've settled out to about 7 or 8% resistance amongst the strains that we're testing. And so, in our guidelines, part of the treatment protocol is to give a gram of azithro. And the question becomes, how useful is that, given that we're seeing much higher levels of resistance amongst gonorrhea strains, and why do we actually have that on our guidelines? The other confounding factor here is that most of our testing is now done by NAT. So previously, in the early 2000s, our testing was done by culture. And because we've switched over to a NAT system, we've really had a decrease in clinical isolates being submitted for culture. And therefore, there's a decrease in the ability to survey for resistance. Now, there's a lot of sentinel sites. There's the GASP project in Canada, which looks at collecting cultures, but we don't have the huge volume of gonorrhea cultures that we used to have because of the molecular testing that's being done. So in 2018, there was a total of 5,607 isolates that were cultured and tested in public health laboratories across Canada, looking at specifically resistance. And, and that's really in the face of hundreds and hundreds of thousands of cases of gonorrhea that were diagnosed within Canada during that time. So we have a very small proportion of cases that we're able to actually test for resistance. So looking at multi-drug resistant and then extremely drug resistant. So for multi-drug resistant gonorrhea, this is really defined as those that have a decreased susceptibility to cephalosporins or have resistance to azithro along with resistance to at least 2 other antimicrobials. And usually that's tetracycline erythromycin. And the proportion of multi-drug resistant gonorrhea increased. So it was 4.5% in 2014, and it was up to 8% in 2018. So that's resistance, to multiple drugs as well as possibly decreased susceptibility to cephalosporins. Again, 2017, a really bad year. Resistance rates were up to 12%. But what you can see over the trend here is that we're sitting at around 8 to 9%. For MDR-GC in Canada. And so, again, a worrying number that may be limiting our antibiotic use in the future. The good news is during that time period from 2014 to 2017, there were only 4 XDR-GCs that were identified in Canada. In 2018, 7 isolates were classified as XDR-GC, so that was 1% of all the isolates that were tested. And this is defined as decreased susceptibility to cephalosporin plus resistance to azithromycin, as well as resistance to at least 2 other antimicrobials. So these are the ones where our standard doses of ceftriaxone certainly and azithromycin would not work at all. This is the only thing that 2017 was good for, was we did not have any XDR-GC in Canada. But what you can see here is that our rate has remained relatively low and stable with 1 to 2 cases and again, 7 cases identified in 2018. So it looks like, at least in Canada, as opposed to the States, we don't have quite as much of the XDR-GC, but we're seeing increasing numbers of MDR-GC. And then overall, what we found in 2017 is that our resistance rate to the cephalosporins still remains relatively low. So decreased susceptibility to cefixime within the isolates that we tested in 2017 was around 0.5%, and that was a small decrease. The same thing with isolates with decreased susceptibility to ceftriaxone, that was about 0.6%, which was decreased from 2.5%. And then azithromycin resistance, really is still on the rise, so 3.4% to 7.6%. And then also they did identify a few high-level azithromycin resistance isolates in 2018. Also in 2018, not surprising, almost 60% of the isolates were resistant to Cipro, so 57.3%. Again, same number resistant to erythromycin, around 56%, and about 47% were resistant to tetracyclines. So what we're starting to really see is a very narrow number of antibiotics that we would use in clinical practice for gonorrhea that will actually be able to work. I won't belabor the genes that are involved in these for too long, because I think there's a lot smarter people in the room who know this more than I do, but there's a couple of mutations that are associated with Neisseria gonorrhea resistance, and that's usually within the DNA gyrase. And there's some high-level resistance mutations that have also been identified. And with the ability to do molecular testing, we are able to come up with tests to be able to do these in the lab. For azithromycin resistance, this is actually important because there are a number of resistance-associated mutations within the 23S RNA gene. And these oftentimes are single-point mutations. This is why azithromycin monotherapy is usually not recommended. And we've seen this with other organisms where we've primarily used azithromycin, is that it's relatively easy for gonorrhea to develop macrolide resistance. And so when you're using azithromycin as treatment, you do see that resistance accumulate very quickly. And then resistance to cephalosporins. So we don't have great criteria for what we say is resistant for cefixime and ceftriaxone, but any MIC greater than 0.5 micrograms per ml would be defined as decreased antibiotic susceptibility. So for a lot of years in areas in the East, so specifically in Japan, they've had a lot of data on cefixime treatment failure. So they were using cefixime as first-line treatment for gonorrhea in Japan up until 2006. And then they had changed their guidelines over to ceftriaxone and actually went with a relatively high dose with 1 gram as a single dose. And then in 2011 was when that first high-level ceftriaxone-resistant gonococcal strain was isolated. And this was from the pharynx of a female sex trade worker in Japan. The story behind this is that she was actually lost to follow-up and didn't complete treatment. And so some of the concern back in 2011 with this was that that strain was not eradicated, most likely, and was still circulating. If we look at the antibiogram from that specific organism, what you can see is that there's, R all the way down, but penicillin was resistant, ceftriaxone, all of the cephalosporins were resistant, obviously resistant to the fluoroquinolones. It had resistance to the macrolides as well as some resistance to the aminoglycosides. So really what you're looking at for successful antibiotics are things like piptazo or meropenem, which we generally don't stock in our STI clinics as our first-line treatment. And this was really the concern when they characterized this organism, that if it was circulating within the population, that all of the first-line treatments that would be used within the clinics would not actually be effective. And they did some modeling with this strain, and basically they found that using that 1 gram of ceftriaxone for treatment, the ceftriaxone MIC of this particular strain would escape eradication in almost everyone. So it'd be very unlikely that the baseline treatment that people would use would actually be successful for treating gonorrhea. And then for cephalosporins for resistance, the most common mechanism is alteration of the penicillin binding proteins in the PEN-A gene, but there's multiple other mutations. As I showed from the history, there's beta-lactamase production. There's also mutations in the MTR gene that can cause overexpression of an efflux pump. So there's many mechanisms by which the organism can become resistant to the cephalosporins. So that brings us to looking at those new CDC guidelines and really going over what the rationale is for that change. So as we've seen, although we do have increasing rates of gonorrhea resistance to many antibiotics, our ceftriaxone and cephalosporin resistance rates for now still remain relatively low, so that should be a successful treatment. So what are the new guidelines? So their recommendations for uncomplicated gonococcal infections of the cervix, urethra, or rectum is ceftriaxone as a single dose. And then they've actually done something wise here, which is looked at the fact that we know that antibiotics are really weight-based dosing. And so that ceftriaxone should be given to patients who weigh less than 150 kilos or 300 pounds. If someone weighs more than 150 kilos, they're recommending a gram of IM ceftriaxone should be administered. And then we've gotten rid of that azithro recommendation altogether. And if chlamydial infection has not been excluded, then you should treat for chlamydia with doxycycline. During pregnancy, you can give azithromycin. And the reason for getting rid of that azithromycin 1 gram is, as I mentioned, really we've got increasing resistance for azithromycin amongst gonorrhea strains, as well as the majority of testing that happens in North America is combined testing for both GC and CT. And if you've excluded CT, you don't need to add CT treatment on. Finally, there's more emerging data about chlamydia, specifically for extragenital chlamydia, that azithromycin as a single dose is not quite as effective as giving doxycycline for 7 days. So that's the reason behind this change. They actually have separate guidelines for gonococcal infection and oropharynx, and that's because it's a little bit more difficult to treat. So antibiotic concentrations within the oropharynx are a lot more variable than they are within the rectum or within the genitourinary tract. So they still recommend 500 milligrams IM as a single dose, and for people weighing greater than 150 kilos, 1 gram of IM ceftriaxone. But really the change here is that there are no reliable alternative treatments for pharyngeal gonorrhea. And they're lining the guidelines, they throw it back on us. They say, you know, if there's penicillin allergy, consult an ID specialist. But really there's, you know, because of those rates of resistance, using things like quinolones or azithromycin, if someone is penicillin allergic in this setting, would really not be effective. So what's the rationale behind these changes? So a couple of really important ones to know. So first of all, it's pharmacokinetics and dynamics. So with ceftriaxone, you need concentrations that are greater than the MIC for about a day. And we know that with a single dose of ceftriaxone, you do get those levels within your body for a day. However, when you do this both modeling and animal models as well as subhuman models, that 250 milligram dose of ceftriaxone doesn't reliably achieve levels higher than the base MIC of the organism. So you're not getting anything greater than an MIC of 0.125 for an extended duration. And so the concern with giving 250 milligrams is that it's really not going to necessarily be effective to eradicate gonorrhea in every patient. When they've done modeling and again, animal studies, what they've looked at is that the lowest ceftriaxone dose that's 100% effective is looking at about 5 milligrams per kilo of body weight. And that corresponds to a 500 milligram dose in a 80 to 100 kilogram human, whereas 250 milligrams only corresponds to about 3 milligrams per kilo per body weight. And so their recommendation for the change is really to try and make sure that we're optimizing the amount of ceftriaxone that's there to eradicate the organism and also decrease the possibility of inducing resistance. The other reason is what we talked about with oropharyngeal gonorrhea, is that the ceftriaxone concentrations, again, are very variable within the oropharynx. We know that treatment of gonorrhea probably requires longer times above the strain's MIC. And so there's a high likelihood of treatment failures at this site if you're giving just 250 milligrams of ceftriaxone. And that was really, again, part of the rationale of increasing the dose of ceftriaxone to 500 milligrams as a single dose. So finally, I'm just going to talk briefly about oropharyngeal gonorrhea before we hand it over. So this is an article I presented at IIDR Rounds, but it's interesting and fun, and I think it's a good one to talk about. So because of NAT testing and in Ontario, you know, we can do the oropharyngeal testing through public health. The prevailing wisdom has always been that oropharyngeal gonorrhea was a result of orogenital contact, and that really it's oral sex in some way that transmits gonorrhea. And then there's been more and more data that's come up that's been questioning that and saying, you know, we can culture a viable organism from saliva. Is kissing alone actually possibly a way that people can transmit gonorrhea? And so there's a study that was published in 2019, which looked at kissing alone as a risk factor for oropharyngeal gonorrhea. And this was specifically a study amongst men who have sex with men. And again, you know, this public health messaging has always focused on the promotion of condom use, even though no one ever uses condoms for oral sex. But really, they talked about just penile anal sex as being the main driver of transmission of gonorrhea amongst MSM. But as I mentioned, there was some data and mathematical modeling that suggested that transmission from the urethra to the oropharynx really represents only a small proportion of those incident oropharyngeal gonorrhea infections that we're seeing. So this was a cross-sectional study that was conducted at the Melbourne Sexual Health Centre, and patients who were presenting for assessment or treatment were asked to complete a questionnaire using a computer, and this was named the kissing survey. It was added to routine clinical care between March and February of 2016 to 2017. So they collected data on the number of male partners in those categories related to kissing. So how many partners did you have where you only kissed? How many partners did you have where you only had sex but didn't kiss? And how many partners did you have where you had sex but also kissed? I'm not sure how everyone can remember that, but apparently they did. And so they had about 3,600 people within the final analysis who had filled out the questionnaire and also tested and had results available for oropharyngeal gonorrhea. So they looked at demographic characteristics, if they were a known gonorrhea contact, as well as their HIV status and their laboratory diagnosis of gonorrhea, took oropharyngeal and anal swabs as well as urine swabs and tested them through that. And then all participants were included in the final analysis if they were male, had completed the survey, and had been tested for oropharyngeal gonorrhea on the day when they completed the survey. So what did they find? So the positivity rate for oropharyngeal gonorrhea was 6.2%, anal rectal gonorrhea was 5.9%, and urethral gonorrhea was actually relatively low at only 2.7%. Almost all the men had kissing with sex partners, most men had kissing only partners, and less than half had sex only with no kissing in the past 3 months. Of the 52 participants, so 1.4% of the sample who had kissing only partners, and that was it. The positivity for gonorrhea in the oropharynx was actually 3.8%. In the anal rectum, it was 2.3%, and urethra it was 0. And for those people who reported sex-only partners, positivity for gonorrhea was a little bit more variable, so 3, 4, and 4%. And the positivity of oropharyngeal gonorrhea amongst the men who had sex-only partners was significantly lower compared with the total study population. So what they calculated was the odds of having oropharyngeal gonorrhea was 1.64-fold or 1.46-fold for men with greater than 4 kissing-only partners and 1.81 for men with greater than 4 kissing with sex partners, compared with men who had 0 to 1 partners in these respective categories. So really, it did appear that there is a role for just saliva exchange, for just kissing in the transmission of oropharyngeal gonorrhea. Obviously, lots more research needs to be done in this area, but it seems that that's a way that gonorrhea is spread. And indeed, from the data in Ontario, what we're seeing is a lot of previously undiagnosed oropharyngeal gonorrhea as we start to screen in that site. So kissing with or without sex may actually be a risk factor for oropharyngeal gonorrhea. Lots of limitations, again, as I said, so lots of people declined to participate. It didn't really measure penile oral sex, and gonorrhea was screened by NAT, so we didn't know if the organisms were viable or not. But again, really interesting data about possible transmission through this method. So I'm going to end there and then hand it over to Gerry. So, important things to know. So for treatment of gonorrhea, higher doses of cephalosporins may actually be required. And I think in Canada, these changes will probably come over the next few months. If you're worried about treatment failure, if you're concerned at all, please send a culture because those organisms can be tested for antimicrobial susceptibility. And as I said, we've got less organisms that we can actually test from clinical specimens to see what antibiotics will work. And then 100%, if you have a treatment failure, please send a culture because it's really important to characterize that level of resistance. So I will hand it over to Gerry.
Okay, so I'm going to tell you a little bit about what's happening, what we have in the pipeline, and to address the problems that Kevin was just mentioning. So I'll tell you a little bit about where things are in terms of clinical trials for new drugs, and then I'll briefly tell you about an effort in our lab to be able to contribute to this area that we started several years ago to look for new anti-gonococcal leads. So the pipeline, you've seen this before. The deal here is that if it's the light color blue is when these drugs are introduced. The dark color blue is when it's no longer useful. And you can see this tends to happen pretty rapidly. Sulfonamides within a decade, penicillin within a few decades, et cetera. And the fluoroquinolones you just heard about, same thing. So we have this challenge where this bug is very, very adaptable. And it's very interesting because it's in the lab, it's very easy to find molecules that kill this bug. But clearly, you know, its ability to, well, they're naturally competent, so they can share DNA very, very easily, and you end up with these mosaic kinds of genome structures, and that results in opportunity to select for resistance. So what's out there, what's available? So our best drug that was there was Azithromycin. This is a ketolide, so this is very much for the chemists in the audience. You'll recognize this is the macrolide structure that's very similar to azithromycin, but with a ketone here instead of another sugar. So these ketolides have been around for a while. And azithromycin been through a few companies. Septra was the one that took it to this trial. It was single dose, 100 milligrams at a time in phase 3. And unfortunately, last year, or 2019, it did not achieve non-inferiority against ceftriaxone and azithromycin. The single dose was in particular important because these ketolides as a class have been associated with liver tox, and so a single dose seemed to make sense. The TPP seemed perfect for this molecule, but because it didn't achieve non-inferiority, this company went bankrupt. And the molecule, as far as I know, is still around. It was bought by Toyoma in Japan, and although I don't see any clinical trials happening now on their website, they say that it's in phase 3 again. So we'll see if that ever gets resurrected. But in the meantime, other companies have been active in this area. So there's, I think, a couple of good news stories coming forward. There's 2 new molecules, so new chemical matter, so the very different than the ketolide that we just saw, which is a derivative of macrolides. One is zoliflodacin from a company called Entasis. Entasis is a spin-off from AstraZeneca. So when AstraZeneca decided to get out of the antibiotics field, it created this new company called Entasis. And this drug is in phase 3 clinical trials, expecting to be to report this year, actually. Another one is by GSK is gepotidacin, and again, brand new chemical structures. And so this is actually incredibly exciting in the antibiotics field. We haven't seen 2 new, completely unique chemical structures emerge in this field for any therapeutic indication, honestly, for decades. So both of them target the topoisomerase, type 2 topoisomerase DNA gyrase, which you'll remember is the target for the fluoroquinolones as well. And just very briefly, the way that these work, of course, is that these topoisomerases are essential for DNA synthesis, because as you're moving the DNA polymerase along the double strand, you create these supercoils, and you have to release that tension. And this enzyme, which consists of 2 subunits, gyre A and gyre B, we don't have to get into the mechanism this morning, but the fluoroquinolones impede this, or they freeze this complex, the whole complex, and cause double-strand breaks on the DNA, and that results in cell death. So both of these new drug classes target the very same enzyme, but in very different ways, which is exciting. So zoliflodacin, which is the compound that's Entasis has come together. It's a spiropyrimidine trione. The spiro means that there's this one carbon that has 4 different substituents. It's very cool. It targets actually gyrase B, a very low frequency of resistance, 10 to the -8. It's not affected by any of this circulating fluoroquinolone resistance in gonococcus. The current trial has a single oral dose of 2 to 3 grams. So it's a massive amount of drug. And again, it reflects one of the challenges in antibiotics is that it's not like blood pressure medicine where you're giving 10 mgs or something. We dose these drugs very, very high levels. So the therapeutic index is always the biggest challenge. So it's in phase 3 trials. This is actually sponsored by GARDP. For those of you who know, it's the British clinical or drug development system that or a sponsor that really is helping bring new drugs to market. So this should report this year. Same thing, gepotidacin. This guy actually binds at the interface of two gyrase A monomers. So when gyrase comes together, it's a dimer of dimers, and it binds at the interface, again in a different spot than fluoroquinolones, causing single-stranded breaks. And here again, notice large doses of compound. And again, as I mentioned before, it's in phase 3 trials now and hopes to report this year. So despite the fact that, as Kevin showed, there is this very significant challenge of drug resistance and multi-drug resistance and even XDR drug resistance in this organism around the world, I think that there's, at least in this field, there's probably some light on the horizon, unless, like azithromycin, it fails to make its non-inferiority endpoint, and then who knows what will happen to these molecules. So stay tuned, but maybe if we're giving this talk next year, we'll have 2 new anti-gonococcal drugs on the market, or at least available to some markets, hopefully in Canada. There's a couple other molecules out there as well. I won't dwell on them. I've looked for some recent data. This is the Famulin, which is a pyromutilin compound derived from a fungus was discovered back in the early 1950s. It's a semi-synthetic version by Nebruvia. And then microbiotics, again, it's building on an old theme. These are spectinomycins, have these drugs that they've reported on, at least at conferences and such. And so, but so far, no additional information that I have. So I'll just take about 10 minutes and tell you a little bit about some of the work that's been ongoing in the Institute, and in particular my lab, looking for gonococcal lead compounds. And the first first objective here is that we were looking for things that, looking for molecules that have some affinity towards gonococcus, that might be new leads for preclinical drug development, or at the very least could become biochemical tools. The molecular biology of gonococcus is very, very interesting, and it's not a lot of people working on it, despite the fact it's a very important pathogen. And just parenthetically, Kevin mentioned, that you saw that picture of penicillin cures gonococcus in 4 hours. Well, there's some discussion by military historians that maybe the reason why penicillin was such an important tool for in the Second World War for the Allies is not because it was great for curing the battlefield wounds, although that was part of it, but actually because they were able to make sure that a good number of the soldiers, which normally would be laid out because of STIs, were actually available for duty, especially during the invasion of Normandy. And so that's really the value of penicillin to the Second World War, not so much on battlefield. But anyways, so this is work that was done by Linda Ejim in my lab, a longtime technician, and Vishwas, who was a Fulbright scholar visiting from University of North Carolina. And basically what they did is developed a high throughput screening platform based on some of these multi-drug resistant and XDR drug resistant pathogens that we had from the Public Health Agency of Canada. And so they developed this platform, and picked in particular this one strain, the WHOY strain, which had high ceftriaxone and azithromycin resistance. And our first goal was to see, could we find molecules that actually rescued ceftriaxone activity, because ceftriaxone being standard of care in many cases. But then our second objective was, can we find molecules that were effective against this strain? So the first part of this is we actually did a pretty large screen against about 75,000 molecules from in the high school screening lab within the institute that were curated to have some advantages for entry into gram-negative bacteria. We screened those at 50 micromolar, and basically this is what resulted. So 75,000 in duplicate. We ended up with about 700 hits. Did dose response on the top 122. Did a lot of work. Most of them ended up being pretty toxic to human cells, but found 4 molecules that were interesting. These are them. Then we ended up reordering them to try and get some dose response. Some of them worked pretty well. Some of them we couldn't get a hold of. And then we wanted to try and develop this program a little bit further, but turns out that the synthesis was really challenging to do. Jake McGolan helped with this, and lots of challenging problems with these molecules. And it turned out that when we looked at the hits a little bit closer, that they were really quite messy, and was really hard to figure out what exactly the active compound was. And so with that frustration in mind, we pivoted away from this synthetic compounds, although there might be some opportunities to go back in that. And that is and then move to natural products, which is something that many of you know has been a focus in my lab for many years. So we have this library of about 12,000 bacteria and fungi that we've collected from a number of different environments. And they all produce all these wonderful molecules. You can see another example here. This is our, I call it our zoo of microbes from all over the place. They all have these lovely different shapes and produce very, very different molecules. And then what we've done is build an extract from each one of these, methanolic extract. So we grow them up, extract all the secondary metabolites, compounds that they produce, and then lay them out so that we can do high throughput screening. We also have been increasingly taking some of these very complex mixtures and partially purifying them in this fashion. So we just take them, grow them, and then do a partial separation on reverse phase columns to generate what we call the prefractionated library. And then you can see that this is actually teasing out a lot of these different, these are all different fractions. So this is one organism. And so normally that would be, the extract would be looking like this, and then you partially purify it. And so you end up concentrating some of these metabolites and partially separating them so you can deconvolute stuff pretty nicely in this way. And so what we did is we did a pilot screen of 10 of or 80 of our strains and found a number that were quite active against gonococcus. So here's one that we followed up on. So exquisitely sensitive MICs against these multi-drug resistant organisms. You can see them here. These are Kirby-Bauer tests. This is our zone of inhibition, and all these different strains. It's really quite good, and we've just published this last month. So this one strain called 1542. So we were quite excited about it. And then this is Venki in the lab, who's a postdoc, and what he found was he purified the active molecule. Beautiful peak here. Excellent production of this compound, and what it was is a known compound. So this is moenomycin. Moenomycin has been known for about 50 years. It's a gram-positive agent, so we were quite surprised to see this activity. Sequenced the genome, found the biosynthetic gene cluster that goes with it. So what moenomycin does is it acts on the cell wall biosynthesis. So recall that penicillins and beta-lactams inhibit the cross-linking and the extension of cells of the peptidoglycan. But the first step is the transglycosylase step. That is, you're bringing in monomers from the inside of the cell and then linking them up to the sugars on the outside. And that's where moenomycin works. So it's a tremendously exciting target. Moenomycin has never been clinically useful because of some pretty serious protein binding problems that you can imagine. If you look at the structure for a second, it's got this big long polyketide tail, so it's pretty greasy, but very effective. So it's been used in agriculture as a chicken feed additive for probably 30 or 40 years, but not in the clinic. But it turns out that I mean, and when we're thinking of what the target product profile would look like, is that if you had again, you want to have a single dose as many times as possible, and so that it's very easy for infected individuals to take, and so they don't have, and follow-up can be challenging. This might actually work really well. So we kind of got a little bit excited about it. There had never been any reports of this molecule in the literature as active against gonococcus. And then we did a lot of work to show that it was actually on target, that actually is inhibiting these transglycosylases, which again is surprising because normally moenomycin has no activity against gram-negative organisms. So it has a unique way of getting inside the periplasm. And the reason why, sorry, just to back up, the reason why it has no activity against gram-negatives is because it can't penetrate the outer membrane. So it does so in gonococcus. And so, we were pretty interested in this molecule. And we've been working with a company that also came at this at the very same way. So we're following up on this with them now, probably providing more backup for their molecule, which is a moenomycin derivative. But I think it's kind of an exciting area to think about moving into. So lots of hits came out of that screen for us. We just did a really small pilot. And so I do think that there's lots of opportunities for us to target gonococcus using natural product libraries such as the one that we have within the Institute. So where are we going with this? We want to continue to look at the structure activity relationships with these synthetic hits if possible. So we need to go back and find out what exactly the active molecules were there. But we're really going to ramp up this screening, I think, in our natural products. I think there's lots of opportunities here to do that, and if we can do so, we can hopefully get to a lead molecule within the next year or so. So that's our objective. We'll see what happens. This is just a team again. We're some remarkable group of people. None of this could have happened without the staff within the Center for Microbial Chemical Biology and all of the infrastructure that we have available there. It really is a unique asset that we have within the IIDR. And for those of you who have yet to use it or get access to it, I would very much encourage you to think about it. This is something that unique to us that no one else has, and I think has the potential to do some good in the world. So I'm going to stop there. Lori, I'm done. Take some questions if anyone has any, and over to you.
Thank you very much to both of you. I'm going to read the questions that are in the chat. So the first question is for Kevin. Dr. Woodward, for rectal GC, do you recommend 500 milligrams ceftriaxone? And with the current treatment of 250, do you recommend TOC for all GC, throat, rectal, etc.?
Yeah, so I think the question that we're going to run into in Canada is, because our guidelines still include azithromycin, is that fulfilling the role that we thought it would, given, if we see decreased susceptibility? So the one good news that we have in Canada, which is separate from the States, is that our ceftriaxone resistance rates are not quite as high yet. So overcoming that with the higher doses may not be necessary. I think, the test of cure, I would probably do for throat if someone is being treated with guidelines and probably is a little bit overweight, right? Like if you're over that 100-kilo mark, the 250 of ceftriaxone may not penetrate the oropharynx well enough to eradicate the gonorrhea. Right now for rectal, I don't think we need to give 500 milligrams if we're treating with the 250 plus the azithro, but I do suspect that that's going to change in Canada over the next year. All the people who would write our STI guidelines are currently very busy with COVID, so some of that delay in responding to what happened in the CDC may be just because of the current COVID pandemic. I think what I took away from the CDC guidelines as well is looking at it's not one size fits all. And so, tailoring your antibiotic to the patient in front of you, especially if they have a larger BMI, is probably the most important thing.
A comment from Martha. She said, I have had more referrals for syphilis in the last year than I've ever seen. And if people are getting syphilis, they're getting gonorrhea. I actually have a related question to that. So do you think, it's a little scary to think about these XDR strains in the US, but do you think the travel restrictions are going to at least temporarily reduce the import of these kinds of strains into Canada?
Yeah, I think so. I mean, most of the gonorrhea resistance that we see in the States, it actually, if you look at the geographical distribution, it sort of starts in Hawaii and moves its way over. And that's because, even as I presented in areas like Japan, Korea, they have much higher rates of resistance to the cephalosporins. So it tends to migrate that way. So probably we're going to see less resistance. I think to Martha's point around syphilis, the problem that we have right now is that access to screening and access to testing is actually much lower. So, in Hamilton, for instance, we have one STI clinic that's open, that's appointment only. They can see way fewer patients because of the distancing requirements. And so that clinic that would see, 70, 80 people walk in who could just get testing done no longer exists. And that's the case in most areas in Ontario right now. And we do know from lots of surveys and things that we've done that people are still having sex, they're just not talking about it. And so there's probably a lot more circulating syphilis, chlamydia, gonorrhea that's being passed from person to person out there that's going undiagnosed. And so I think, post-COVID, when all these things open up, I think we're also going to see a large increase in the number of STI diagnoses that probably were missed during the pandemic.
Okay, thank you. A question for Gerry from Brian Coombs. Can moenomycin penetrate eukaryotic cell membranes, or could it be modified to? Thinking about a magic bullet for both Neisseria and chlamydia.
Fascinating question. I have no idea. So we should test it, Brian. I guess I would put the question back to you: is that if chlamydia don't really have peptidoglycan, would moenomycin even work on them? Although I guess it's debatable whether or not that's a good target in chlamydia. Let's put it that way.
I think there is evidence that chlamydia does have at least part of the peptidoglycan machinery that it uses for replication, or not for growth, but for cell division.
Yeah. So yeah, I know that, but I guess the question is it enough of a target? So it would be great to try to test it for sure. Let's do it. Okay.
Any other questions from the audience? Feel free to type them into the chat. So a question from Mike Surratt for Gerry. Great talks. And Gerry, is moenomycin active against Neisseria generally, or just gonococcus? And is resistance present in poultry?
So I don't know about other Neisseria because we didn't test it, but it's an excellent question. I would assume yes. So no resistance in poultry. It's very hard to get resistance. We tried hard to get resistance to moenomycin. It's really well known to have a frequency of resistance well down to the 10 to the minus 10 to the minus 11 because of challenges and with that target. So yeah, excellent question. very little resistance. This makes it a really good lead molecule.
Okay, thank you. I actually had a question about this single dose idea. You know, I understand the rationale for it in terms of follow-up, and because the people getting these diseases can be difficult to track down. But it just seems crazy to me that we expect an antibiotic to work in a single dose when that is clearly a problem. So would either of you like to comment on the single dose aspect of this?
So, I think your comments around the difficulty in getting people is probably where some of it originated. The other thing is that really when gonorrhea was thought as simply an organism of urethritis, you didn't need a long time of antibiotics to clear it. And then when extragenital sites like rectal and pharyngeal came in, we just had that idea of giving a single dose and that should work. And that's part of the reason why I think going up to the higher dose makes more sense. Because you will have a much longer time with the antibiotic present above the MIC. Getting those patients to come back for multiple doses is difficult, and there's really not, because of Cefixim's variable bioavailability and distribution, there's really not a good oral option where you could say, take a week of this, like we can do with Doxy for chlamydia. So I think, again, going to that higher dose may solve the problem. I don't think anyone's done the research with rectal and pharyngeal of what is the optimal dosing strategy to actually eradicate it every time.
John Whitney had a question for Gerry, which he has answered in the text, but I'll read it out anyways. So is moenomycin effective against other medically relevant gram-negative cocci, so Moraxella and Haemophilus?
Yeah, and the answer is we haven't tested it. It's a great suggestion to give it a shot.
John has a second question. Did your experience on this project further refine your views or opinions on the utility of natural product collections versus synthetics? As a non-expert, the latter seems fraught with reproducibility issues.
Yeah, I think at the end of the day, it's all about your library. And I think this library has been problematic for us. We selected, we call it our gram negatives library. It was pre-selected, cost us a lot of money to curate, and it was really done, I think, properly. But it turns out, I think the chemical manufacturers, a lot of them have amines positively charged, and that's hard to do on a large scale, I think. And so, at least that's my opinion. I think Eric's probably had some similar reproducibility issues with this specific library. It's one of the reasons why him and Jake are working together to build, their own really high quality sort of gram negatives library based on those kinds of the current understanding of the rules of these things. And so it's a lesson learned for us for sure that we need to check purity of these molecules. Of course, the natural products are totally impure. And so, it's a flip side of that, because they're, as you saw, they're really complex mixtures, and it takes a fair bit of effort to be able to purify the single compound. So that's why, and we could even try to do those kinds of things, and have done for some of these compound libraries. It's just, that's not the reason why they're supposed to be. That's the reason why they're so expensive, is because you're supposed to have a pure molecule. So some of it is buyer beware on our part.
Okay, I'm afraid we are out of time. So I want to thank both of you again for a really interesting rounds, and thank our audience for joining us. I hope you have a good day.